So we're going to switch gears a little bit here and go into some of the tools and some of the things that SWAMP and CCHAB have been working on to try to help if water managers, if counties, if other areas are having Cyano-HAB issues and you don't know what to do or you need some assistance with some of these things. So I mentioned earlier, my name is Bev Anderson and I'm with the SWAMP unit at the State Water Board. And I have been working on the Cyanobacteria program that SWAMP has been funding since, well, for about a year or so. Also working very closely with the CCHAB, the California Cyanobacteria Harmful Algal Bloom Network. And we are doing a lot of stuff. There we go. So I want to go give a little bit of information. Did I go one too far? No, I didn't. About California issues specifically. We heard this morning that from Dr. Carmichael that cyanobacteria or blue-green algae are found in most kinds of water bodies and they are found worldwide. They can exist as single cells, they can exist in colonies, they can form blooms that cover entire rivers in large areas. In September last year, Ohio had a bloom that was 600 miles long. And this is our very own Klamath River where they can experience blooms that are up to about 200 miles. So these things can end up being massive. And of course, Clear Lake, I'm seeing nods from some Clear Lake people, the entire lake can pretty much be covered by cyanobacteria blooms. So some of the blooms are relatively harmless, although they can be unsightly and cause some issues with that. They can have taste and odor chemicals associated with them. But some release various kinds of toxins, and you heard a lot about the toxins this morning. I'm going to quickly go over a few of those toxins again that we've been dealing with. Some of these toxins can cause skin irritation, liver damage, and in the case of antitoxin A, they can cause very rapid onset of symptoms and death in dogs and livestock, and we've had a number of dogs that within a half hour to an hour of exposure have been killed. So it's a very difficult thing to deal with. There's a lot of issues going on that we need to try to sort through as we're getting some of these programs going. And just about every day I come into work, there's something new that comes up. So we are dealing with it as it comes along. We've also got some non-cyanobacteria species that are starting to cause blooms. Those, again, were mentioned this morning, the Primnesium, the golden alga, and there's a few lakes in Southern California that have had those and caused massive fish kills. And late 2015, there was a lake in northern Nevada that golden algae just completely wiped out their fish stock that they had put into a lake for some kind of a fishing tournament. So those can also cause some serious issues. Freshwater HABs have been increasing in frequency and severity worldwide. And we're also seeing this kind of pattern in California. These increases have been variously linked to increasing temperatures associated with climate change, high nutrient loads that support the bloom growth, and then this past couple of years in California with the recent drought that's been linked to the cyanobacteria blooms that we've been seeing increasing because we've got a lot less water flow, lakes are low, rivers are low, flowing slowly and warming up, and these are some of the key triggers that cause some of these blooms to occur. At this point, we're not entirely clear what's going to happen for the rest of 2016. We are already seeing blooms. We're getting calls and emails about blooms, largely in Region 5, which is up the Central Valley, but Clear Lake is having a bloom. Some of the East Bay parks are blooming already, so we are starting to see them, and we don't know exactly what's going to happen for the rest of the year. If we heat up with the rains that we had this last winter and spring, that may have washed a lot of nutrients down into the water bodies that have been piling up in the hillsides for the last few years of the drought. We'll see. We're just keeping our fingers crossed and hoping that it's not crazy. But we're there. Cyanobacteria generally originate and bloom in freshwater bodies such as lakes and wetlands. However, blooms can also start in rivers and streams, and we're having rivers and streams that have cyanobacteria that we wouldn't normally characterize as blooms. Benthic cyanobacteria are releasing toxins, so we're seeing some of these occurring. The Eel River, Klamath River obviously, the Russian River. We had a potential cyanobacteria issue in the Sacramento River last year. It wasn't confirmed, but we're not sure what happened there. But we are seeing them in different areas where we had not seen them before. Another thing that can occur is rivers and streams can act as conduits for the cyanobacteria and or the toxins. So this morning, Dr. Carmichael mentioned the sea otters in Monterey Bay. Those were tied to a cyanobacteria bloom in Pinto Lake, about 9 kilometers away. So again, a lot of issues going on. And we're dealing with some things that are happening all the time. Microcystis is the most common cyanobacteria that you hear about. This is the one that we're always looking at. People, if they hear about cyanobacteria, they are thinking about getting tested for microcystis and microcystin, which is the toxin microcystis produces. Microcystis can produce other toxins, and microcystins can be produced by many other different cyanobacteria. So it's a very complex system where if you know what species or what genus of cyanobacteria you have, you don't necessarily know what kinds of toxins it'll produce, and vice versa. The microcystis is one that has had some triggers and health levels created for it. In 2012, OIHA developed a recreational human health action level of 0.8 micrograms per liter. And then just last year, the US EPA set a drinking water human health advisory for microcystin of 0.3 micrograms per liter for little kids and 1.6 micrograms per liter for older kids and adults, which is proving to be a difficult thing for drinking water to deal with. How do you tell a family that, no, your toddler can't drink that, but yeah, the 6-year-old, So they're trying to figure out how to deal with those kinds of levels. Luckily, in many cases that we have here in California, the levels are in the thousands to hundreds of thousands micrograms per liter, so we don't need to worry about whether it's 0.3 or 1.6, right? So, silver lining. These are the three main toxins that we're dealing with, and these are the ones that recreational human health triggers have been worked on by the CCHAB, and this is just to give you an idea of how complex the whole cyanobacteria to toxin issue is. You can see microcystins can be produced by a number of different species of cyanobacteria. Likewise, anatoxin A, which is the one that we have identified as the dog-killing one, and livestock and other organisms as well, also produced by a number of different species. And then Cylindrospermopsin, which we don't hear an awful lot about, but the US EPA developed drinking water triggers for that. So it's been included by the CCHAB for recreational human health levels. And that's also produced by a number of different genera and species of cyanobacteria. And then, to make things even more interesting, there are a number of other toxins that can be produced by some of these same organisms. Saxitoxins, you heard quite a bit about this morning, and they can be produced by some of those same Anabina, Phanozomenon, and Cylindrospermopsis can produce saxitoxin. And a paper that was published last year in 2015 by Betty Fetzer et al. describes the results of a study using spat bags, and they detected toxins throughout California in rivers and streams. They found that in about 1/3 of the streams there were toxins being detected, and there's Betty has indicated a preference of having saxitoxin be tested for because of its prevalence in some of the areas throughout California. So these are some other issues that we may need to deal with. Yes?
Would there be any differentiation between Northern California and Southern California with respect to what you last said?
As far as the streams?
Saxitoxins.
I would have to look back at the paper. I think they were pretty much finding it in most areas, but I would have to look more carefully at it. I could send you the paper if you like.
Right. The reason why I ask is Ray Cordella keeps insisting that we don't need to search for
worry about
those because
saxitoxins.
we'll never see them in Clear Lake. I just wondered if you...
Well, she was detecting them more in rivers and streams, and that's what their study was. So I can't really speak to saxitoxins in Clear Lake, but I would suspect Rafe is probably more of an expert than I am on that. So I would not be terribly worried about it at this point. Lori, do you remember if she was getting saxitoxins more in south or north? Or did you have a chance to look carefully? Okay. So they're out there.
It's definitely rivers and streams.
Definitely rivers and streams at this point. Okay. And then really quickly, some of the hot spots that we have in California, I've mentioned Klamath. The Klamath Basin has been getting cyanobacteria blooms for many years. Clear Lake, obviously, they just love cyanobacteria. And we've got some beautiful samples of Clear Lake water here that you can take a look at. The San Francisco Bay Area and Delta has been getting cyanobacteria blooms for a number of years, and there are a number of studies in the Delta looking at what they're getting. And then Pinto Lake, Monterey Bay, and Southern California is now getting those Primnesium or golden algae blooms. And we're now starting to see them throughout the state water project. So pretty much the entire state is going to have to be dealing with cyanobacteria at some level or another. And then I just want to go really quickly into a couple of these areas where we have hot spots and we've been dealing with cyanobacteria blooms for another year. There's some work being done with satellite imagery, and Clear Lake has been a lake that has had a lot of satellite imagery. The satellite was Maris, and I'll talk a little bit more about that. This satellite produces images that look at different wavelengths of light, and they can determine if there's chlorophyll in an area in a lake. They can determine if there's phycocyanin, things like that. And these are older images from the Maris satellite. And this one shows in, I think it was 2013, this first one, clear, not much going on. It's a nice blue lake. And then in July of 2011, you can see we've all been brought up to know that red means bad. So this is indicating that there's a high level, and I believe here they were actually looking at chlorophyll, but it's showing that there's a massive bloom of something, and that this has been linked to cyanobacteria.
Well, the left picture is from February as well.
Yeah, February
July,
2013, right.
Very
right.
different in terms of cyanobacteria blooms.
Right. And then this was the Pinto Lake issue that I mentioned. You know, Pinto Lake is way up here. They have blooms of microcystin quite regularly, or microcystis, sorry. And then several years ago, sick and dead otters were being found way down here on the beach in Monterey Bay, and eventually they had 30 dead otters, and the number may have increased since then. And that was linked to the microcystin coming down from Pinto Lake, and this image over here shows the jaundice that develops when the liver stops functioning properly, and these otters had this jaundiced appearance to their tissues. And then these are the East Bay parks. In 2014, East Bay Parks had 2 toxic cyanobacteria blooms. Lake Temescal was closed to swimming for half of the summer, and Lake Chabot, the lower one, had 3 dog deaths. In 2015, they had a number of additional lakes that had blooms. A different Lake Chabot up here in Vallejo had a dog death. All of the lakes that allowed swimming in here that had blooms had to be closed to swimming. And in the original Lake Chabot, this one down here, they had a high of 150,000 micrograms per liter detected in their samples. Remember, the human health trigger is 0.8. So these are the numbers that we are dealing with in some of these areas. So what is SWAMP doing? In response to these statewide blooms, SWAMP started to develop an assessment and support strategy and is funding a lot of infrastructure that's being developed and some of the tools to help with the successful implementation of the strategy. The goal of the strategy is to have a program to assess, communicate, and manage freshwater HABs. And we're working with the CCHAB to develop these things and to create the coordination that's needed to get this program moving in the right direction. And this document, this strategy was released in January of this year. Was it January? Oh, was it that long ago?
Yeah. January.
You'd think I'd know that. So the Freshwater HABs Assessment and Support Strategy has a fairly neat framework here, and this is how it's set up. It's divided into 3 main components: a response to HAB events, ambient monitoring, and risk assessment. And in most cases, what people who are dealing with HAB events are going to be interested in is this section here with the response to HAB events. So an immediate event response would start with a response to a satellite imagery hit for phycocyanin or other evidence of a bloom, and would inform public notification and bloom management, and that would all fall under this. The updates to the 2010 Blue-Green Algae Voluntary Guidance document fall under this immediate event response, and they include a flow chart of the process from initial identification of a bloom through posting, toxin testing, things like that, and I'll go into that in just a little bit. Long-term event response would include things like setting up local action plans or getting TMDLs in some areas. Pinto Lake has a TMDL that they're using to help get the funding and programs set up to try to deal with their HAB issue. So those are some options for long-term event response. And then the strategy also calls for regular monitoring of water bodies with recurrent blooms and for those at high risk of blooms. At a higher level, a statewide or regional level, we've got development of the infrastructure that's needed to do all of this stuff. And SWAMP has been funding some of that, a good portion of that. Monitoring at a state and regional scale, and a risk assessment at all scales. So if we start looking at the infrastructure consists of 7 components. And SWAMP is funding some of this. SWAMP is funding the download and analysis of satellite imagery to notify water body managers of blooms and to try to evaluate temporal trends. Those satellite downloads will also help for developing quarterly newsletters and bi-weekly bulletins that we can send out to water body managers or anyone who's interested in receiving that information. We're also working on a centralized website and reporting system where we would have data storage, and the data is going to be stored in the Water Board CDN database. But this website will allow for visualization of the data that's there and access to the data.
Can I ask you a question?
Yes, ma'am.
So because CDN doesn't communicate with the water quality exchange, how does, how do you make decisions, or what's happening with data that tribes are collecting, or maybe federal agencies that are collecting, that are going into federal databases?
We are trying to figure that out because all we can do at this point is draw from CDN. So we're working on trying to get as much data as possible into CDN, and then I believe at some point in the future the two will be communicating both ways, but it's not going to be
It's going to be years, from what I understand, right? I mean, because they're working on CDN being able to, they're working on
CDN to go into WQX,
but not CDN.
but not the other way. Right.
Right.
So yeah, we're working on trying to get access to the data that's needed from these other.
Well, I think one of the things that could help is the regional data centers. I mean, I've been communicating with the Central Valley Regional Data Center, and they are probably maybe underfunded to take on new projects, but you know, we're willing to submit our data through CDN, and it's pretty much the only data that's on Clear Lake is data that we're collecting.
Right.
So it's pretty important to get somewhere.
Yes.
So, but we've been installed in the Central Valley Regional Center just because of potential lack of funding and whatnot. So I don't know if there's anyone in particular at the state who's working on this, but it's just, I would like to talk with them. Because they said if you put it in CDN, then there's going to be a duplicative effort, or you know, or eventually they're going to be talking, and it's going to cause a
Right. If it's
big mess.
entered into both, it's going to end up doubled up in
Right.
WQX.
Because it's in WQX right now.
So yeah, Lori, I'll put that on your.
I'm just trying to think if we could either get the portal to also
To
pull
draw
data
from-
from WQX, or try to get the data.
I thought
from
that was,
I could talk to our
I
CDN.
thought that was happening.
Yeah, we could call it when it,
Yeah.
yeah.
You thought it was happening?
But-
I thought that that's what's, they're working on crosswalk.
Yeah. Well, yeah,
But that-
yeah, that's.
But that's to
No,
have
that's to go
CDN
CDN
go to
to
WQX.
WQX, not CDN
Oh, right.
to draw from WQX. So we would have to get our system to draw from WQX somehow. So we'll have to have that conversation. But yeah, thank you.
Yeah, we can figure out a way.
Okay.
We'll get it, because we know the Clear Lake data is, I mean, you guys have been doing it for years, and we just don't have access to it at this point.
Right.
So, We need to.
We have extraordinary numbers.
yes, I've seen some of your numbers.
Not as high as Pinto Lake. My goodness. Oh yeah, looks right.
Okay, so while we're waiting for a water quality, My Water Quality portal to be developed, SWAMP has put up a web page where we're going to be putting in a lot of the tools that we've got, and then those will be migrated into the water quality portal. This is up and live at this point. If you go, and things are slightly different now because I made some changes to the website after I dropped this into the PowerPoint, but there's an algae identification thing here, and if you go to that, it'll take you to identifying soft-bodied algae of California, and it's got pictures in there. We're working on getting a key, like Wayne talked about this morning, that dichotomous key system into that. So that will be available, and I'll be adding some of these documents that he was talking about this morning, the USGS identification, things like that. Also, if you go to the reporting forms and just go to the Bloom reporting form, we've got a nice Google form in there that will walk you through if you see a bloom or you know that your dog is sick because he was drinking water where there's a bloom, you can go into that form and just fill it out, and it will fill out a spreadsheet that comes to me and a couple of other people, and we can monitor those things. And if there's something we need to get contact you about, we can do that, but we can start monitoring some of these blooms.
Is that information going to be given to all our veterinary offices?
Yes.
Because the problem with what you're doing is so few people know what you're doing.
Yes. And that's why we're out doing this stuff, and we're going to keep getting out and doing these presentations and getting this information out to people. So, and then also here under publications, there's the HAB strategy, which is this with all of the information about it. And you'll notice down here I've got a link to the California Cyanobacteria Network, CCHAB. That will go directly to the CCHAB page and all of the documents that they have. And that link is also up here under publications. So you've got different ways to get to the information, and as we start adding tools, there will be more of those links available. So if you go either here or to the CCHAB webpage, you'll be able to access all the tools that we have out there for you.
Hey, Bev, I just want to make a comment to what she said is, as we do press releases as well, when we find out that there's cyanobacteria blooms and we're notifying, like say, environmental management departments, and they decide, the public health and them decide to do a press release, we're providing these links to these websites. So as Bev goes about and tours and stuff, as we continue to have problems, this information is going to be making its way out to the public because it will be included in press releases.
Yeah. So yes, that's a big part of what we're doing, is trying to get the information out to the public as much as possible.
So why are the press releases on it only going to environmental managers then?
Well, the press releases are press releases that local entities are putting out and sending to us.
Oh, I thought you said there's some things that are coming from the state as well.
No, what she said was if someone's putting out a press release, she sends them this information.
So do you
So that
recommend?
they know that.
Do you recommend press releases on with certain thresholds like the OIMA threshold, or what CCHAB has come up with?
That's coming out of the CCHAB, and yes, there's a subcommittee that's working on communication and coordination, and they will be, over the next several months, coming up with fact sheets for veterinarians, for physicians, and we'll be looking at doing some sort of standardized press releases with that information in there. So yeah, that is coming out of the CCHAB and moving forward, hopefully soon.
Can I just make a plug from EPA's point of view? I don't know if you remember the slide this morning, it was the nation that Dr. Carmichael had of the whole United States and the number of incidents. Those are coming from press releases that are on the internet, so the newspaper. So to the extent that if you do a press release, hopefully they'll get captured, but it'd also be wonderful if anytime you do a press release, you can make sure that that you let people know through that webpage so that we understand what's going on in the state and we can be responsive as much as we can and be aware of what's happening.
Yeah. And I'm looking at the time and I really need to speed up. So I'm going to talk fast. So I mentioned the map that is going to be going up onto the website being developed right now. We're actually looking at having two sort of map interfaces. One would look something like this, where if a lake manager posts a lake for some reason, they've got cyanobacteria bloom, they've detected toxins, they can post. So in this case, these are examples. Don't worry, don't panic. Clear Lake's not totally shut down, and neither is Lake Berryessa right now. But these are examples. So if Clear Lake did have high levels of toxins, they could post on this website and say, okay, Clear Lake is in a danger toxin level, but you'll notice with the slashes, that indicates that there's a partial closure. Lake Berryessa down here, wow, there we go, is indicating that it's under a warning level. So based on the triggers that CCHAB has developed, this would be a warning level, and that would be a full closure. And then if you click on those lakes, it'll give you more information about what's going on. So that would be one of our lake, or one of our map interfaces. The other one would actually be drawing those satellite images. So this is an actual satellite image from Maris satellites, and I'm not sure what year this is from. We've got 2002 to 2012 for that. Again, this is Clear Lake. And then if you click on the lake, it'll zoom in and draw up the data from CDEN that's available for that. So this is showing data from CDEN. In this case, we're showing the lake biomass mean and lake biomass median based on the satellite imagery. And you can clearly tell that your big issues are here in this part of Clear Lake. And then nothing's going to happen. There we go. And then you can also go a little bit deeper and you can find out where samples are actually being drawn. So these little purple diamonds are where samples are being drawn in this case.
As Meoni's report.
And from there it'll draw information, the data from CDEN, and we'll work on trying to get that also from WQX. And if you're a total numbers geek, you can go to this data table tab up here, and you can get actually all of the data that the satellite has produced. And you can draw all of the data that is coming out of CDEN at this point. So, and you can download that if you want to get all of those data. So these are a couple of the map systems that are coming all along and hopefully will be usable within the next, I don't know, several months. Okay, and then other parts of the infrastructure that are being developed, we've got a number of event response guidance documents, and the first of those is the voluntary guidance updates that CCHAB has been working on. This is a very important guidance document that we're getting out in pieces. We got 5 portions of it out earlier this year, and we're starting now to work on some of the other parts that need to be updated. These can all be accessed on the CCHAB network webpage at this point. And I'm not going to try that because obviously you won't see what I see if I go to the website. But down, if you scroll down on the CCHAB webpage, you'll come to this resources section, and that will give you all of these updates down here. The original 2010 document is accessible as a link, and then the updates that CCHAB has been working on. And those include this, which is a decision tree to walk you through what to do if you have a bloom or if you have a suspected human or animal illness. And you can see it's as simplified as possible, which means that it's almost impossible to get good information out of it. So that's why we have an associated narrative to go with it. So if you go in and you have a bloom, you have something that you suspect is a bloom, your first step is asking, are cyanobacteria or cyanotoxins tested? And if you go to the narrative, it'll tell you, how do you know this? And it'll give you step-by-step stuff. Very difficult to go through the decision tree without having the associated narrative because there's a ton of nuance to what's going on with that. So you want to make sure you have all pieces. And then another thing that has been updated is the trigger levels. So I mentioned the 0.8 was the OIMA recreational human health advisory limit for microcystins. So that's what we have as a caution level with the CCHAB, and then warning and danger levels. So there's associated signs that can be posted that go with these different levels. Anatoxin A, you'll notice here, it's any detection is considered a point where you need to put up a caution sign, and that's because of the dogs. Because dogs tend to do things that hopefully humans don't, like lick gooey green stuff off of their fur. and eat smelly stuff off of the beach. So they can tend to get very high levels of toxin, even if the actual toxin level in the water is not very high. So hence we're recommending, or CCHAB is recommending, that if there's antitoxin detection at all, put up a caution sign and ask people to keep their dogs out. Still probably fine for everybody else to go in there. You know, little kids might tend to eat that yuck. But generally, adults, young children, they're going to be swimming in the water, they're going to be splashing around. Low levels of antitoxin are not going to be a huge issue, but you want to keep the dogs out. Okay. And then cylindrospermopsin was added by the CCHAB because of the EPA addition of cylindrospermopsin for drinking water. So we put it in there as well and developed some trigger levels for that. And then the other thing that has been updated is some signs. So we've got 3 levels of signs that are being recommended: a caution, a warning, and a danger sign, with associated information about what that means as far as whether you should swim or eat fish or what you shouldn't do, things like that. And we're working on getting forms of these signs available on the website that can be altered so that if you've got a water body where dogs aren't allowed, you might want to take off the icons that say keep your dogs out, because you don't want to encourage people that, hey, obviously dogs are allowed in and we're telling you now to keep them out. So we're working on getting that a little more editable. Currently there are forms of these signs on the CCHAB website that you can edit in contact information, so you can put in local health officers or whoever needs to be contacted if something's going on. Yes?
So these are kind of like we have fish advisory, fish consumption advisory, where we, like State Board, Regional Board, wouldn't necessarily post them, but
Correct.
they are for
Correct.
the water purveyor,
Yeah.
lake manager to use.
Yeah. And all of this is voluntary. So we're providing the tools and giving people the information they need, but it's really up to the local to decide whether they're going to post these signs and do this stuff that's being done. And there are some situations where some of the locals in various areas are saying, you know what, if we have anything going on, I'm putting a sign up. And they're putting a caution sign up and just saying, okay, we're giving people the information they need, they can deal with it as they do. Some areas are saying, if a dog dies, I'm going danger, period. So that's a local decision that needs to be made, and that's not something that we would step in on. Yes?
Bev, could you make these parts, these individual bits of these signs, individual bits, so we can make up our own sign based on all the individual bits?
That's the discussion we had just yesterday. So yes, we're working on that.
Yes.
Yes, we are working on that, yes.
I would just point out for the group that some of the things that Bev is showing have been updated since this was put
Yes,
together.
they
So
have.
go to the webpage, don't use these for the most up-to-date information on the posters and the thresholds and things.
Well, they can't use these ones because they're on my PowerPoint. So yeah, if you go to the website, you will get the most up-to-date ones. Okay, some other things that are going to become available and some of the things that may be of more interest to you guys. SWAMP has contracted to have standard operating procedures and health and safety protocols written. We are expecting that within the next month-ish or so. So that will give you those step-by-step. I've got a bloom at my lake. Where do I sample? What do I do? What kind of bottles? All of that information. Yes?
And will this come with a hands-on training?
We're working on that. And also along with this, we've got another contract to get lab SOPs written as well. That one's been kind of held up with contracting, but that'll be coming out hopefully later this year as well. So all of these things are coming in so that we can get data that can be compared. Currently, when we see data, even on CDEN, We don't know what it means because different labs are testing different things. They're testing different congeners of microcystins. Some have total, some have certain congeners. So it's really difficult to make some comparisons. So we're hoping that if we can get some SOPs out there, we can start getting comparable data.
Are you doing anything to encourage more laboratories to go into this analytical work that have never done it before? Because we have a problem.
Yes, we do.
A big
Yes,
problem.
we do.
Because the amount of sampling that we're all going to be collectively doing tomorrow, where are we going to send it all?
That's a good question, and I have a list, and I didn't bring them up here, but I will get them.
But it's a very, very, very short list.
It's actually not that short, but some of the labs are definitely a ways away. So it includes labs in Florida and various other places. But yeah, it is a problem. There's not a lot of analytical capability in California.
Is there an organization that these laboratory owners are a part of that you could go and give a presentation to and tell them, this is going to be your next big breadwinner?
Not that I know of, but I'll look into that. Or I'll have our lab person, Marissa, look into that. But she does have a lab network that she's been working with, but I don't know outside of that if there's something beyond it.
CTAC is a good stab.
Okay, I can't.
Well, also, UCMR 4 is, is going to include this in the test, so things are going to develop.
Sounds good. But we do need the capacity. Okay, and now I really need to speed up. So a few other things that are going on. We do have some funds at WPCL with Fish and Wildlife to do some toxin analysis. So that is available if something happens, if you have a dog death, if you suddenly have a bloom and you have no idea what to do. If you contact me, we can hopefully get some testing done. And working closely with Sue at EPA, we can get some, they can do some microcystin. We're working on anatoxin, cylindrospermopsin, so we can get things tested that way. Training and education, well, you're here, so you know. Last year we put on 4 of these trainings in which I had only like 15 minutes worth of stuff to say. So there's been a lot of stuff developed since then. This year 4 more. We did 1 last week, this week, and then there's 2 again next week to cover various parts of the state. And then, as Carolyn mentioned, we're hoping to do something later on when the SOP is done on some field sampling and go more in depth into the tools that we have available and spend some time out there with people doing that. Where am I here? There we go. Applied research and outreach are 2 important areas that have minimal funding. We're working on some of those. As I mentioned, we have that satellite imagery. What I didn't mention is the satellite that we're dealing with right now is a pretty low resolution satellite. So the only lakes that we can actually get information from are pretty large lakes, like 250 acres, which still covers, we've got 255 lakes on our list. And if you think about one person having to take in the information if it comes in, that's a lot. But there are about 3,000 lakes in California, so we're missing 90% of the lakes out there, essentially. And there are a couple of other satellites that are available that have much higher resolution. So there's some work being done to be able to access and download data from those higher resolution satellites, and we're working with Rafe Cadella at this point to start doing some work on getting the algorithms written for those. So hopefully we'll be able to access the data from those higher resolution satellites. And also something that came up this morning is the citizen pilots. I think that came up during the discussion. Oh, that was at lunch. Citizen pilots. There are some amazing pilots out there that just want to go out and fly their planes. So there are groups that organize these pilots and get them set up with appropriate cameras, and they will go and fly areas and just take tons of photos. And you can get those data from the pilot. So Eric has been sort of spearheading getting that all working for us. And likewise, some citizen monitoring programs that I'm going to hit on, and I will be less than 5 more minutes. A few other things coming along. Where is this? Oh, phycocyanin came up this morning. There are some areas that are starting to use phycocyanin probes, and this is a phycocyanin probe and data out of Ohio. They are starting to look at just putting the probes out in various places in Lake Erie and other lakes around Ohio, and they're finding very good correlation between phycocyanin levels and what were they looking at? They were looking at cell counts. So phycocyanin concentrations vary based on the type of cyanobacteria and the turbidity of the water, so it's not perfect, but they can use the phycocyanin probes to evaluate trends, and they're definitely seeing some good correlation. So they're looking at how they can start using the probes rather than doing constant sampling in some areas. And then a very exciting thing that's starting to come up is using QPCR. Wayne went in a little bit into the genetic analysis this morning. We have secured a couple of QPCR machines for California. We've got one at Region 1 where they're starting to do some pilot work to see how the genetic testing matches up to toxin levels, cell numbers, things like that, to see if they can use QPCR in what they're looking at. And I just was able to swing a deal to get one for WPCL, so we'll have another one in Sacramento, so we can start looking at using QPCR. This is another instrument that Ohio is really pushing forward. It has been mandated that all of their water agencies run QPCR. Well, they take samples and send it to the one lab that does it at this point until they've certified other labs. And they're, again, looking at replacing a lot of their discrete sampling for toxins with QPCR. The nice thing about QPCR is that you can test for the genes of several different toxins all at once. So in the case of this particular company, they have the primers to test for microcystins and nodularin, cylindrospermopsin, and saxitoxin. They're currently working on anatoxin A, but they don't have it. And that came about because this company originated in Australia where they don't have anatoxin A. So they never bothered to deal with developing it. But since he's been coming and doing a lot of work in the United States, about every time he's talking to people, the first question is, do you have anatoxin A? So he's now working on developing that. And there's actually another company in Oregon where they do have an anatoxin A primer. So this is an up and coming, inexpensive way to test a lot of potential toxin producers very, very quickly. And the basic premise is, if you don't have the gene, you won't have the toxin. So there are definitely some drawbacks to this. If you're in a situation like the Pinto Lake, Monterey Bay area, if it's just the toxin being carried down, QPCR isn't going to detect that. It's detecting the genes that are in the cells. So, you know, there are issues that come with this. If there's just a toxin, you're not going to get anything. So, yeah, there's issues with all of these, but we're working on the best way of using the tools that are available and providing them in the toolbox.
Ballpark, how much does that cost, that equipment? You just
Ooh.
bought one for...
No, I didn't buy one. I
Oh.
sweet-talked one.
I think I've heard people say, depending on the size and the quality, it's like 20 to 40,000 for a full setup.
Something like that, yeah. So they're not cheap. But when you're dealing with Australian men, there are ways to get there.
Mine's on loan right now at Region 1
Yeah.
from Mark.
So yeah, they're essentially on long-term loan. And that's the same as the one that'll be going to to WPCL, it'll be on a long-term loan. So, and if they make good use of it, the term will go longer. So, and then just really quickly, a couple of other things that are available on the internet. This is one of the documents that Dr. Carmichael had in his Dropbox for you, and I've got a link here. You can quickly go to that out of my presentation. There's the website that I tried to go through out of ours. Okay, so the soft body stream algae of California, as I mentioned, that link is right in that SWAMP Freshwater HABS page, so you can go there. And this cyanoscope, I think Eric is going to go over the cyanoscope a bit over at the lab.
I'll have it out for people to come see me.
So this came up a little while ago. You can join into this cyanoscope on iNaturalist, and if you get a picture of cyanobacteria, you can upload it, or you can just go to this site and compare it to the amazing photos they have on this site and see what they are. Photos that get uploaded, eventually someone can get in there and they will verify them, and you can see when you go into the site, if you go to the photos of the organisms that have been uploaded, some of them will say, I think it's research grade or something. Those have been verified. We know, an expert has looked at those and said, yes, this is what it is, this is the species. Others are people thinking, this is what I've got. So, you deal with that at the level that you need it. Another potentially useful tool, Colorado put out a very simplified guidance for drinking water managers, telling them what to do step by step to deal with an issue with their drinking water facility. And then I mentioned the laboratory list. This is online, and I actually have enough copies of it, hard copies, that I can give this to you. These are all the labs that we know of, what they do, what their turnaround time is, who to contact, things like that. Do they do ELISA or LCMS? All of that information. It's all just in a really super neat spreadsheet for you. And then this again is one of the documents that Dr. Carmichael had on his links this morning, and it's amazing. If you haven't looked at that, go in and look at it. The micrographs are amazing in this, and it's free to download. And that's it.